Hey there, it is Mr. W from ScienceMusicVideos.com, and I am so happy to be here for another live YouTube Performance AP review session. Still figuring out what exactly to call that, and still figuring out how to get my earplugs to look pretty good on the screen. And I think I got that figured out with this move right here. So let me say this. I am gonna do this, and here I am. I was just checking on another monitor that I have that I'm actually here, and I'm wondering if some folks could actually just like say hello, tell me who you are, tell me who's here. Let's hear a couple of people just go into the live chat and tell me who's here, who's doing what, so that I know that I got some folks out there that are ready to do some learning. So anybody here? Really looking forward to seeing some people. I see 19 people, 17 people, a couple of people are giving me likes. Just maybe just type your name in the chat. And fantastic, Amelia. So great to see you. Fantastic. Who else is there? Just tell me who you are and maybe how you learned about this chat also. Did I email you? Did you learn about it through, you saw it on YouTube? Did your teacher tell you about it? So I'm hearing Jiangmen, Ariana, Lindsay, Aiko. This is so fantastic. So many of my students and other students too. That is so fantastic. Anybody else? Just tell me a couple of other things. Chaticus, good to see you again. Squidcat, Kim Grace, Ben, fantastic. I am so happy to have all of you guys here. This is really great. And so for some of the folks who aren't my students, will you just tell me, just put in the chat a line about how you learned about this live event, because one thing that I really want to do is I want to improve my outreach to folks, and I want to make sure that I'm doing a good job getting these lessons to everybody who needs them. So Iman is here, fantastic, and Kaya, my students are showing up. This is so fantastic. This makes me really, really happy. So anybody though who's not my student, just want to just give me a quick line about how you learned about this. Anybody? Just one? All right, I got a Nintendo boy. It's fantastic. People are saying hi. That's great. You know what? I think that what we're going to do is maybe I'll ask you that a little bit, but right now I think it is time to move into the lesson. And so what we're going to do is we're going to start with a little bit of music, and we've got protein synthesis. Oh, and I got Squidcat, and let's just see what we got here on my other screen. I got that, and all right. So guys, if you go to ScienceMusicVideos.com, and what you can do really easily is, let me go right here. Thank you. Okay, if you go to ScienceMusicVideos.com, you can go to, there's a contact, etc. link. And then if you click music videos, and then if you go to protein synthesis, you can get all the words to the protein synthesis song, which we are now gonna sing together. Are you guys ready to do this? I gotta move myself a little bit over here. Yeah, now that's a good position for this. And let's see if this actually works. We're gonna go and do some singing right now. Protein synthesis. Let's make it happen. And oh, that's a little anticlimactic. Oh no, I'm getting the okay, let's go back. Let's try this again. Let's see if I can bring up this video this time. And it is not happening. You know, sometimes what happens is you practice over and over again, and you expect the software to actually work, and the universe just does not do it the way that you want to. We're going to try this one more time, and even though you practice, and it ain't happening. So maybe we'll do a song another way. another time. We got some great music. Oh, I have a lot of people saying let's jam. I'm just wondering if I could figure this out. Let's see if I can do this. And let's go here. Let's go here. Let's go here. Nah, this ain't gonna work. Guys, you know what we're gonna do? We're just gonna start learning some biology. So let's do this. Let's go here. Let's go to the next screen. And here we are. Okay, so here's what we're gonna do today. I'm gonna move myself over to the right side, and here I am, and there, perfect. Okay, so we're gonna do some practice FRQs, and I just wanna let you know that you've got a practice FRQ solution at my website, sciencemusicvideos.com. It's the BIOMANIA AP Bio test preparation system. You can access it on sciencemusicvideos.com, and you can access it on your phone. It's a fantastic solution. It's lots of practice, and that, of course, is what you need to be doing. You need to be doing a lot of practice, reading, and writing. And so here's what we're going to do. Just going to let you guys know that these questions that we're about to do come from the FRQ portion of BIOMANIA. You can see on screen how the settings should be showing you what you would do with the FRQs. You're just going to choose units 1 through 6, and that's because units 1 through 6 are the only units that are going to be on this year's AP Bio exam. So just choose 1 through 6. The questions come at you randomly. You get a question. The job on your part is to think about it, to write out the answer, and then to check it against my response. Nintendo Boy is asking me how much it is, and if you're not my student at Berkeley High School, it is $9.99 for the app and a subscription to the entire website, which gives you access to the entire website, including tutorials about every topic in AP Biology, plus the BIOMANIA system is $19. It's the best bargain in biology learning. So let's go on right now and let's do a practice FRQ. But before we do that, I want to say, as good as my stuff is, you cannot, under any circumstances, skip working with the best material that there is, and that's the material that's straight from the College Board. And the way you get access to that is you go to AP Central, you search, or you just Google AP Biology exam questions. What you've got there, you can see over my shoulder, you've got questions from 2019 and back. This page happens to have 2018 and back, but 2019 and back, and you've got scoring guides. And you should study all of those topics that involve units 1 through 6. And how are you going to know which ones those are? Well, I've set up a study guide for you, and it's right here. And basically, you can download this. This is your BIOMANIA practice journal. I'm going to put the link for this in the comments below, or you can find it on sciencemusicvideos.com and basically download it. This schedule on this page up here, this table on top, is going to tell you what to practice and when. All right, it's going to tell you specifically what FRQs to do, what units to be working on BIOMANIA, and Pranav, it actually isn't really like that. And Nintendo Boy, again, it's $19 for a subscription to the website. It's $9.99 for the app. And let's go ahead and let's do some reviews. So here we are, and let's do some. I wonder if this isn't going to work. That's going to make me really sad. Music is not working for me. None of my videos are loading. How did that happen? Who knows? But what we're going to do is some AP Bio review. I'm seeing something starting to happen, but it's not really working, and we're going to do this another time. Next time we do a review session, all my music and videos are going to be working. It's going to be fantastic. All right, we're going to do practice FRQ number 1. Now, what you're going to see is going to be a lot of text on the screen, and that can be kind of tough. So if you want, what you can do is go to sciencemusicvideos.com, click the link for Mr. W Live, and you'll see this question underneath today's video. So let me walk you through it. You got a diagram on right, and it represents a portion of the electron transport system in the inner mitochondrial membrane. So you know about this system, we're going to review it, but this system transfers electrons from NADH and FADH2 to oxygen. Energy from this from this reaction is coupled to pumping of protons. Now, one thing that's going to happen on this year's AP Bio exam is that they're going to be throwing scenarios at you, and then they're going to show you an interference with a biological system. And that's what's going on here. So here basically you've got two drugs, drug X and drug Y, that interfere with certain aspects of the electron transport system. So what I want you to do right now is this. I want you to just think about what's going on. I don't want to see answers. I don't want to see anything. I just want to have a sense of you out there doing some thinking about how you'd respond to this question. It has two parts. Right now, we're just going to do the first part, okay? So everybody, think about this question for a minute. I'm going to be super quiet, and I'm seeing a question from Rhea, who's saying that I thought there were going to be no visuals like diagrams on the FRQs, and you're saying, oh, I hope that that's the case. I don't think that's the case. I think it's actually very likely that there's going to be diagrams. Visual representations are a big part of AP Bio. I haven't heard anything about there being no diagrams, and I am almost certain that there'll be some experimental data. So there's going to have to be some graphs that you're going to interpret. There's going to have to be some tables. And I'm seeing some other people who are asking for glycolysis. Maybe if I can figure out how to get sound to come out of this thing, I can do that at the end. But right now, here's what I want you to do. Everybody, I'm actually going to have to sing a song a cappella since my accompaniment is not going to work for me. And let's do this. This is the mitochondrial electron transport chain. And one thing that I am going to do is I'm going to move myself over here. And I hope you know this song, because the chorus of the song teaches you almost everything that you need to know about the mitochondrial electron transport chain. And let's do this. I'm going to sing it a cappella since the music's not going to come through. It goes like this. The mitochondrial electron transport chain uses electron energy for pumping protons from the mitochondrial matrix to the intermembrane space, increasing proton concentration in that place. The only way the protons can escape is through a channel and an enzyme ATP synthase, which uses diffusing protons kinetic energy to make ATP, to make ATP from ADP and P. So I kind of flubbed that at the end, but this rhyme basically tells you what you need to know about the electron transport chain. So here's what I want you to do. This is the electron transport chain. I'm going to show you this again on another slide right here. Okay, so here's what you've got, and I'm hoping you can see my cursor, all right? We've got NADH that drops off electrons to the electron transport chain, and that uses these are proton pumps. And basically, in the same way that like when you plug in a device, like your computer, and I'm going to move myself over to the right side so you can see this full diagram. Okay, so in the same way that like when you plug an electrical device in, the electricity is used to do some kind of work. Well, basically, NADH is kind of like it's an electron supplier. This whole system is actually kind of like a fuel cell. And what it's doing is it's providing electrons that flow through the electron transport chain, and they use that electrical energy to pump protons from the mitochondrial matrix to the intermembrane space. And those protons accumulate in the intermembrane space, and then they diffuse through ATP synthase, which is simultaneously an enzyme and a channel. And the power of their kinetic energy is used to create ATP, because ATP synthase can catalyze the formation of ATP from ADP and inorganic phosphate. So I've got a couple of questions, and I want to see the answer in the chat. Everybody ready? Ready to do some typing? Okay, so here's the deal. Which process will drug X immediately disrupt? Put your answer in the chat. Let me see it. It's a number. 1, 2, 3, 4, 4. So what's the answer? It's a number. Let me see a number in the chat. Let's go for it. The first interactive part of this exercise. I'm not seeing a single number yet. Let's go. Guys, it's just a 1, a 2, or 3, or a 4. It's like a little multiple choice quiz. Let's see some numbers coming in. Oh, seeing a 2. All right, that's good. Shivam had the first answer. Not saying if it's right or wrong. I want to see some more answers in the chat. Let's go, guys. I'm going to use this as an opportunity for a little... Seen a lot of 2s. A lot of 2s. A lot of 2s. Okay. And even though I saw a lot of 2s, you know, I got to say something. You know, I get stuff wrong all the time in class. It's not impossible that I'm going to get this wrong now. But I actually am pretty sure that the answer here is not 2. It's not 2. It's 1. And here's the reason why. And of course, it's about interpretation. So this is showing how drug X is keeping these electrons from going to the electron transport chain. Drug X is intercepting those electrons as if a quarterback is throwing a pass. Like, you know, NADH is the quarterback and it's throwing a pass to the electron transport chain, but there's an interception happening. So those electrons are not going. So now I'm seeing some people starting to get this right. It's a 1. It's 1 that's really the answer, because these electrons now aren't going to flow into the electron transport chain. Drug X is like positioned over here, right? It's keeping these electrons from flowing to the electron transport chain. And now that I've told you that, I want you to think of some consequences. Think about what the electron transport chain does, both immediately and long term, and think about what some consequences of this interception of number 1 is going to do. So everybody think about this, and no holds barred. Right now, put it in the chat. Tell me what some consequences are of this interception. I want to see some answers in the chat. Let's go, guys. All right, we know it's number 1. Okay, anybody have any ideas about what some consequences will be of this interception? Okay, I'm actually just going to look, and I'm going to just stroke my chin over here. People are putting in answers. I see an answer from Naomi Johnson. Good job, Naomi. I'm not saying it's right or wrong, but good job responding. Everybody's doing some thinking. Ariana. Jiangmen. Okay. More detailed answers coming in. Fantastic. Fantastic. I'm seeing some really great answers here. Okay, and Jiangmen thought of one that I didn't think, but now these answers are pouring in. This is fantastic. All right. All right, guys, I'm going to tell you the answer now. This is fantastic. I need to move myself over to the left side. Let's see if that works. There I am. And here's the answer coming in. Okay. Well, first of all, what we're going to be doing is we're going to be breaking off our electron flow to the electron transport chain. So there's no electron flow. This whole system won't be powered. It's as if we pulled the plug on the entire electron transport chain and the whole system is now shut down. Now, the consequence of that is therefore we can't power these proton pumps. You can't really see this one over here, but I'll just put my cursor here. We can't see this proton pump over here. And excuse me, you can see this proton pump over here, and no proton pumping is going to be happening, and the same thing here. So we've shut down electron transport. We've shut down the pumping of protons, and if there's no protons, now there's no longer an electrochemical gradient for creating ATP. Remember, the mitochondrial electron transport chain uses electron energy for pumping protons from the mitochondrial matrix to the intermembrane space. None of that's going to happen. That is all over. So basically, we've completely shut down the process. Okay, now let's move on. Make sure that's set. Go. Okay, so now what I want you to think of now is why is another interceptor. I think that's a cornerback in football. So drug Y. What will drug Y disrupt? And I'm going to move myself so you can see the screen again. Here I am on the right side. Okay, so process Y. The answer is 1, 2, 3, or 4. What will process Y disrupt? Will somebody please type something into the chat? Want to see some answers? Seeing some good answers coming in here. Keep it coming, guys. You're doing a fabulous job. Okay. Anybody else? You guys are fantastic. How did I get so lucky to have such fantastic viewers watching my live stream? I don't even know how this happened. There was a pandemic, I had a studio, I set up my live stream, and all of a sudden I have all of these students who are putting in the correct answer, because indeed the correct answer is number 3. It's number 3. And let's talk about what the consequences of that are. What's the result of blocking electron flow to, I sort of have a little error here. It's really the result of blocking electron flow to oxygen, and that means I kind of gave away the answer. And what's amazing is that you can be a teacher and you can work for hours and hours on a PowerPoint and you still make errors. And there you have to stand in front of your students and they point them out. And now I'm just seeing them myself. But guys, here's what I'd like to see. Lucas, you put 8, but 8 wasn't even one of the choices on the screen, but I'm glad you're thinking in a big way. Good job. Fantastic. So here's what I want you to do. I want a sentence. And you can't really see the question. I've got to move myself. I'm going to go here. So what is the result of blocking electron flow? Blocking electron flow of drug Y. What's the results? I'm seeing some answers coming in. I kind of goofed up on this question. Chaticus. Good job. Simi, nice. Jiangmen, good. You put an asterisk next to your response. Oh, I see. All right. All right, guys, I think that's enough. Basically, the only consequence of drug Y is that there will be no production of, no consumption of oxygen, and no production of water. No consumption of oxygen, no production of water. But the rest of the electron transport chain should be working pretty well. In other words, you're still pumping protons, you're still generating an electrochemical gradient, and that means you're still going to be able to power the cell's activity through the creation of ATP. So that is the first part of this question. So here's what I'd like you to do. I'm going to move myself again. And I'm going to move over to the right side. And guys, you know this. You totally got this. So what I'd like you to do is I'd like you to predict whether cells will continue to consume O2, cells will continue to produce CO2, what will happen to the rate of ATP synthesis. Really, all we've been doing is checking for understanding from what I taught you just before or what you figured out by yourself. So let's just see some summaries in the chat just so that I know enough to know that you're getting it, all right? So again, what we're talking about is drug X over here, and we're saying you got to make some predictions, and that's a big part of what you're going to do on this year's AP test. So let me see some predictions coming into and put it all together in, you know, a nice, well-designed sentence. And I'm just going to calmly wait until those answers show up in the stream. Seeing some answers come in. Just get a couple of more people typing into the chat. Okay, people are doing great. Guys, here's the first response. You're predicting whether the cells will produce O2, and no, because there are no more electrons that are flowing towards oxygen. Now, I know there's a little lag time, but here's the deal. I want a justification. Many people have said, See, I'm pretty sure that the cells will continue to produce carbon dioxide. And here's my challenge to you right now. Why? Why? What processes are continuing to occur that will produce oxygen? No. What processes are continuing to occur that will produce some carbon dioxide? Why will these cells produce carbon dioxide? Why? Why? Somebody in the chat, please. Shivam is asking me how long this is going to go for. I was planning on an hour. Anybody know why carbon dioxide will continue to be produced? Can you perhaps give me the names of some processes? Yes. Fantastic. Guys, the way I'm reading this, carbon dioxide will continue to occur because the Krebs cycle and the link reaction, which preceded it, are going to continue to occur. So with Krebs and link, then we've got the two processes that create carbon dioxide. Now, rate of ATP synthesis should decrease. No more proton pumping to power ATP synthesis. For those of you who are extra sharp, who are like sharp as, I was going to show a pencil, but I don't have a pencil, so my whole prop idea just fell apart. There's not going to be any ATP production because there's no proton pumping, but there might be a little bit of ATP production, and the reason why there might be a little bit is that there will be some ATP that's produced during the thing that you asked for, glycolysis. Glycolysis, okay? So guys, that was fantastic, and now we're going to do the second part of this. But second part's going to be pretty easy, so let's just go pretty quickly. Okay, so drug Y. Will the cell continue to produce oxygen? Will the cell continue to produce carbon dioxide? What will happen to the rate of ATP synthesis? And an explanation. Let's see it in the chat. And Shivam put something about photorespiration. And photorespiration is something that only happens in plants. And it's a process that interferes with photosynthesis. So I've got to move myself so that you can see this question, and I'm kind of getting the hang of this. There I am. So what I'd like you to do is respond to these 3 questions. Let's go, guys. I'm taking a little sip of throat coat tea. Lucas, you're thanking me, but I'm not really sure what you're thanking me for, but I appreciate the thanks. Okay, I'm seeing answers come in. Okay, guys, I'm seeing enough answers. It's time to do this. Share you with some of the answers. All right, so we're not going to produce oxygen anymore because we've intercepted the flow of, we're not going to consume oxygen anymore because we've intercepted the flow of electrons to oxygen, which means that we're not going to basically reduce oxygen to water over here. So that process is done. Okay, will the cells continue to produce CO2? Absolutely, because of Krebs and the link reaction, so that's not affected. Rate of ATP synthesis, I think it's going to stay the same. You still have proton pumping that's powering ATP synthase, so it really shouldn't be impacted. And now we've got our explanations. So guys, here's the plan. I don't think it makes sense to do this right now since we've just gone over this, but what I want you to do at some point is come back to this question and try it again. Write out the whole thing. This question is in the BIOMANIA app, and by trying it again, by writing it again, you can get another try. So let's switch gears a little bit and let's do something really fun. Oh, I wanted to just first of all inspire you, say way to go, you guys are with me on this. And now, anybody have, let's save this for the end. Let's save this for the very end, okay? So we're gonna do a biology quiz. Everybody ready? Biology quiz. Answers go in the chat. Everybody ready? With my classes at Berkeley High School, I like to have people stretch out their fingers a little bit before they do what we call finger biology. And of course we can't do that. We're just relegated to this little chat window. That's all the feedback I can get from you, but I'm very happy to get that. It's a beautiful thing. Hey, I'm actually wondering if somebody might do something. You know what I'm noticing? I am noticing that right now I've got 32 viewers. How does everybody feel about taking out their phone? Isn't that funny? I'm asking you to take out your phone. I would never do that if it weren't a pandemic and invite somebody. Invite somebody to come to YouTube, to my channel, or just to go to Science Music Videos. Let's see if we can get these numbers up from 32 to much higher numbers. So everybody, before we start this quiz, invite one person, one classmate, one friend of yours who's taking biology, and invite them into the chat so that we can see. Invite them into this quiz, invite them into this YouTube extravaganza so that we see tons and tons of people coming in here. And again, I'm totally grateful to each and every one of you who is here right now. And I've got about the same number as I would have if I were teaching a class. I'd love for there to be, I don't know, 60 or 100 or 1,000. Wouldn't that be so cool if I was teaching 1,000 people at one time, and if we had 1,000 people reviewing for biology, even in the midst of this COVID-19 pandemic. So, Shivam, I'm having a little trouble with the music, so I can't do glycolysis right now, but Next chat, I promise I'm going to solve that. I thought I had it all nailed, but we'll figure it out later. So, guys, everybody ready? Everybody, I hope, texted somebody, invited someone else to come in. See if we can get our numbers a little bit higher. The number went to 32, but let's see what's going to happen now. You're going to type your answers in the chat. Here's question number 1. mRNA carries information from DNA to a... What is it? What's the answer? Let's see it in the chat. Come on, guys. mRNA carries information from DNA to a what? I'm going to cultivate a little tranquility while these answers come in. I'm seeing one. I'm seeing more. Yes, fantastic. You guys are all nailing it. Guys, you couldn't make me happier. I'm as happy as a ribosome. Ribosomes aren't happy. Ribosomes are just particles. But I'm extremely happy to see all these right answers coming in. Guys, let's do something else, okay? How about this? All right, ribosome's the first answer. Next question. If you're my student, you're probably going to nail this one. Enzyme activity is based on blank between the shape of the enzyme's substrate and the shape of the enzyme's active site. What is the answer? What is the answer? Please fill it in. Spell it right. Let's see. Just waiting for some answers to come in. This is the asynchronous nature of internet teaching. Seeing it come in. Seeing these answers come in. I got one. Just one. There's more. Ben, Lucas, Lindsay, Shivam, Squidcat. Squidcat, what kind of name is that? Jadicus, Naomi, Amelia. You guys are fabulous. Fabulous again. Pranav, Aiko. All right, next question. You guys ready? The answer indeed was complementarity. All right, complementarity. Hand in glove. People in love. Codon and anticodon. So many things in biology. Complementarity. All right, let's go again. Now, next question. During blank blank, tRNAs bind with specific amino acids and bring them to the ribosome. It's a 2-word answer. Put it in the chat. Let's see it. Come on, a 2-word answer. Put it in the chat. Let's go. Squidcat, I'm sure your name is wonderful, but anyhow, we'll worry about that another time. Anyhow, you want to be, that's great, Squidcat. So, but let's see some answers. Some answers to this question. BIOLOGY. Some answers to this question. BIOLOGY. You guys are just killing this. You guys are fantastic. All right, guys, indeed, it's a 2-word answer. If you said translation, you're smart. You got this, but, you know. All right, protein synthesis. 1 or 2 more. During the Krebs cycle, a series of enzymatic reactions produce ATP through substrate-level phosphorylations, transfer electrons to electron carriers, NADH and FADH2, and release what as a waste product? Let's see the answer. seeing answers come in. Let's get a couple of more. All right, guys, the waste product is indeed carbon dioxide. And is that it? Okay, last one. This one's so easy. I shouldn't have even bothered with this one. In fact, I'm going to skip it. Electron transport chain. All right, now let's go to this one. What kind of alleles separate as a result of crossing over during meiosis? Okay, it's one word. Some kind of allele can separate as a result of crossing over during meiosis. What kind of alleles? Guys, I'm looking forward to some answers to this question in the chat. They're going to be there in seconds. And then we're going to move on to our second FRQ. Okay, this is very interesting. Now, as a teacher, I know enough to know that if almost everyone is getting the answer to a question wrong, I probably asked the question badly. But guys, all right, everybody go like this, okay? These are homologous chromosomes, all right? So like my fingernails, those would be like homologous alleles. It doesn't really make sense to think that those would separate as a result of crossing over during meiosis. No. So like, let's say, I'm actually going to try and do this. I don't think this is going to work. Oh, the problem is I can't really write with my left hand. Okay. Okay, so homologous, all right? Big A, little a, right? They're not going to separate as a result of crossing during meiosis. What would separate would be like if this was an A allele over here and this were a B allele over here. So what do you call alleles that are on the same chromosome that can separate as a result of crossing over during meiosis? What do you call those? Guys, what kind of alleles separate as a result of crossing over? It's, I think I lost everybody. Yes! Finally, a right answer popping up here. It's not homologous alleles that separate. Homologous alleles, you know, they might sort of jump different spots, but they wouldn't separate. It's linked alleles. Linked alleles that can separate as a result of good. I'm seeing it coming now. Linked genes, of course. Now you're seeing the right answers. Linked. Good, Lucas. Ben, fantastic. All right. So, guys, I was originally planning on doing 2 FRQs, and I think that would put this session as being like, I don't know, it took 45 minutes just to do this. So I think that what I want to do is this. First of all, let's skip that. We'll do that next session. Okay. So here's the question. I'm going to open this wide up right now, and I'm going to go to this view. over here. Does anybody just have any questions, like something you're wondering about from biology that's come up during review that you want me to try and answer? Anything? You know, I can do this. Or at the very least, you can put the question in the chat, and I can promise to get back to you. But you know, anything that you're curious about that you want me to respond to, any biology questions that you're looking for answers to, you know, we can sort of think about this as a kind of a virtual. Shivam is asking for singing again. Shivam, all right. Let me see if I can do this. All right. I gotta do a couple of things. Sound. Okay. That might have been what I needed to do. Am I still okay? Are you still hearing stuff? Okay, I'm gonna try something. I'm gonna close this PowerPoint. I'm gonna go like this. I'm gonna go like this. Drag this over to this other monitor. Guys, sorry about this. I didn't expect to be having these technical problems here today. Thought I'd worked all this through. Okay, that's good. Now, let's see if I can go like this. Ooh, making some progress. Okay, let's see if I can go like this. Okay, still got everybody. Let's go here. Let's go me over there. Oh, we're making a lot of progress here. Let's go here. And what I was going to do was do this and music videos. And let's do what I had planned. And let's go to protein synthesis. No, that's not the right page. Here, music videos. And here, protein synthesis. And let's see if this works. Holy cow, it does. Guys, we're going to do this. Okay, guys, I want you to join with me. I want everybody singing at home, okay? Is everybody ready to do some singing with me? All right, I'm feeling like I've maybe lost my audience here. Let's see. All right, so let's go here. Everybody ready to do some? All right, listen, Lucas, here's what I'm going to do. I'm going to do this one song, and then I'm going to answer your legit question. Let's go. All right? Everybody, let's sing. Look at what's coming out of the nucleus, destined for a ribosomal strand of mRNA. It's got the code, the information for the protein we'll be making today. The RNA message is organized into codons. That's a sequence of bases 3 that spell out one and only one amino acid, a genetic code that works universally. A codon has its match in an anticodon, 3 bases on tRNA's bottom side. tRNA's job is to bring amino acids to ribosomes so proteins can be synthesized. mRNA, tRNA, and ribosomes plus amino acids make a protein, make a machine. Read the codon, make the polypeptide, that's translation. Cells synthesize a protein. The small subunit of a ribosome binds with an mRNA's leading end and slides until it reaches start codon AUG, indicating where translation begins. A ribosome has 3 binding sites where tRNAs combined. The A site's first, P's next, then E, and the E's got an exit side. The first tRNA in this translation prices on the start codon and binding site P, and that tRNA carries an amino acid that goes by the name of methionine. And now to finish this initiation, the ribosome needs to be made complete. The large subunit binds a small subunit. Now we're ready for translation to proceed. mRNA, tRNA, and ribosomes plus amino acids make a protein, make a machine. Read the code on, make a polypeptide, that's translation. Cells synthesize proteins. Now we're ready for the next step, elongation. An amino acid carried by a new RNA with an anticodon matching with the second codon binds at the site called A. Then the ribosome carries out an enzymatic action binding these amino acids with a peptide bond, so a dipeptide hangs on the A site RNA. Watch translation move along. Next, the ribosome shifts exactly one codon over in a move that we call translocation. So the A site's empty and the P site's loaded, and the first tRNA's in the E location. E site tRNA makes its exit, and the next tRNA fills a site called A, and the ribosome catalyzes away. In your cells, it happens every second, every day. And watch that polypeptide, see it get longer as each codon gets translation. Ribosomal robums RNA instructions as proteins get created. mRNA, tRNA, and ribosomes plus amino acids make a protein, make a machine. Read the codon, make a polypeptide, that's translation. Cell synthesize protein, protein synthesis, translation. Protein synthesis, translation. Protein synthesis, translation. Protein synthesis. We started with initiation, which was followed by part 2 of translation, elongation. Now it's time to finish synthesizing this protein with a finale called termination. When the ribosome gets to a stop codon, there's no tRNA anticodon that matches, so A site's bound by a release factor, the polypeptide detaches. tRNAs of P and E sites float away, ribosome breaks apart, no longer one, the polypeptide folds into a functional protein. Translation is done. mRNA, tRNA, and ribosomes plus amino acids make a protein, make a machine. Read the codon, make a polypeptide, that's translation. Cells synthesize protein, protein. Named ribosomes plus amino acid make a protein, make a machine. Read the codon, make a polypeptide, that's translation. Cells synthesize proteins. Protein synthesis, translation. Protein synthesis, translation. Protein synthesis, translation. Protein synthesis, translation. So good. Guys, you see Max Cowan's name on the screen? Max Cowan, he has a band called Attikid. The guy is a genius. He's the producer of all my music. Okay, guys? Oh, I'm seeing all of these emojis. That's like emoji overload. That's fantastic. Okay, so I'm going to look at some real questions right now. Gitanjali Rao, is it open Google or just open Notes? So, they're going to have, I think, what's called a lockdown browser. And the lockdown browser is going to, let's go here. The lockdown browser, is that going to be able to keep you from looking at your phone or a book or something like that? I have no idea. Like seriously, I think we're all kind of in uncharted territory here. But I don't actually think it matters, because I actually think the idea of you looking at a book and getting meaningful answers, it's not going to be like that. They're going to be throwing complex questions at you. I mean, a little bit like the one that I threw at you right now, but like more complex with a complex data set. And you're going to have to be able to look and understand the data and then explain the underlying biology. That's what it's all about. This isn't the kind of thing where like texting someone, looking at a book, that is not going to help you. You've got to know how to handle data. You've got to know underlying biology to be successful on May 18th. There's no two ways about it. And that's why what I'm having my students do, and what I'm hoping you're doing, is just, writing FRQ after FRQ, looking at different data sets, looking at those College Board questions. Because the ones that are on AP Central, that's the real thing. You got to look at those. So that's the question. And how detailed do your answers need to be? Squidcat, you're answering that on the AP exam. They got to be really detailed. They've got to be really detailed. And at the same time, it's going to be approximately like 25, 30 minutes for one and then like 15 minutes for the other. I forget exactly how it breaks down. But, you're going to have to be able to write with detail to get your score to a 4 or a 5. And, I don't know what it's going to be like this year, but I do know that nationally, every year, only about 7% of test takers wind up getting a 5. Now, my students do much better. And the reason why they do much better is I think it's because I run them through a good practice session, and I have a good comprehensive AP curriculum. And this, again, is why I totally want to encourage people to be using, my BIOMANIA test preparation system, because it'll give you the practice that you need so that you won't really be surprised when you're there on May 18th. And that's the goal. And again, remember that you can do this on your phone through the BIOMANIA app, or you can do it on the website. So, Lucas, you said that you had a question before. I saw a bunch of emojis. Okay, Jatan, okay. I'm seeing a question from Shivam: how to cover biology section, sir, for pre-medical entrance exam. Shivam, I'm really in the AP Bio niche, but I actually think that if you prepare yourself for the AP Bio test, you're going to be preparing yourself for most exams. You might have to do some additional studying, but that's basically what I'm thinking about. And okay, Marianne, that's a very, very nice comment. Ria, how to prepare notes for the exam. I actually don't think notes are going to be really, really useful. I think having really significant content knowledge is what's going to be useful, and that's why I'm going to encourage you to do what's on the screen over here. I think that if you go through my FRQs, if you go through my multiple choice questions, and if you go through the flashcards, you will have the content basis so that when you go into the exam, I'm not going to say you'll know everything, but you'll have a good shot. And like, I don't think that preparing tons of notes ahead of time is really the strategy. Studying ahead of time. Gitanjali, you're using BIOMANIA. Should you focus on the FRQs? Yes. I would say that the priority is this: do the FRQs, do the flashcards, do the multiple choice. Those should be your priorities. The thing that I like about the multiple choice, and the reason why I'm having my students do the multiple choice, and everybody knows, my students, this is required, is that the multiple choice is really the only part of my system that's kind of like not honor system, but it actually is checking for understanding and giving you feedback. So if you really want to get to a high level of content knowledge, again, do the multiple choice questions. It's so easy to do. You just go here are multiple choice. I even have a little instructional video. You'd start. I'm going to go to my student identity, student login. And once the thing loads, just do this. Select questions 1 through 6, and just those questions are just going to come at you randomly. And actually, by the way, that's a much better way to study. Don't just do the thing where like you're going to master topic 1 and then, unit 1, and then go on to unit 2. It's much more effective to get it all at the same time, because that's going to help you develop the interconnected thoughts in your mind. Let's see more questions. Okay. Libby, that's a great comment that my song saved your exams last year as well as this year. And Simi, about what score you need for a 5, I have no idea. 2 questions. There used to be like all these formulas, and what was the book? There's like the Princeton Review is really big on like figuring out how to game the test. You know what? I don't even know what these books are like. So forgive me, Princeton Review folks, for saying that. I'm sure these books are wonderful and get you all this study and learn a ton of stuff, just like my stuff does. But I don't know what score, and I don't tell my kids to do that. What I tell my students to do it is just learn a ton of biology. Learn a ton of biology. And again, one way that you can do that is by using my BIOMANIA preparation system. It's an app. It's also on the web. Unfortunately, if you're not in a teacher's class, you got to buy it. That's just the way it is. But it's not very expensive. Get your parents to buy it, and that's it. Bryson, how often do I do these live streams? This is really like the second one that I've done. I don't know. So I think I'm going to do them at least once a week. This is actually an interesting question. Maybe you guys can give me some feedback. I don't know how many people I have left here. Let me check. Oh, I have almost everybody still here. Can you tell me how many of these would you do per week? So the answer is like, say I did 3 of these per week, and I'm thinking like I would do them. I'd do 2 afternoon sessions and 1 evening session. Can everybody put like a 1, a 2, or a 3 in the chat right now? How many of these would you do? Can you please give me some feedback about that? Because if you're, if there's a demand for these, I would do more. I would do more. So 1 session a week, 2 sessions a week, or 3 sessions a week. Let me see some responses in the chat right now. It's going to help me schedule this. I see 3 cups from Squid Cat. I'm assuming that means 3. Two. Grace is saying 2. 2 or 3. 2, 3. Three, 2. Okay. 3. Okay. So guys, it's past 3, and I think we should bring this to a close. I just want to show you a couple things on my website. So over here, you see this live question of the day? So I am going to put schedule of Mr. W Live's screencasts. So I've got my next one scheduled for Thursday. And then, so let's start with that. And here's what I would like you to do. I want you to help me publicize these. I'd like to get more people doing this because there's 250,000 kids who are going to take the AP exam. And, I think that this kind of thing could be useful. So I want you to spread the word about this. Now it's going to be Thursday, the 16th, 2 p.m. Okay, so there it is. Spread the word. Tell everybody. I'll be back here 2 p.m. Pacific time with another thing. If you've got some suggestions about what would be super useful for me, then I'll tell you what I'd like you to do. What I'd like you to do is, see right up here on the top, it says contact. See that's contact over there? Shoot me an email. Say, hey, Mr. W, I really liked this. It would really be great for me if you did more of this. This is a topic that I'm having trouble with, and I'd like more help with this, so on and so forth. So, guys, please spread the word. I'll see you on Thursday. This has been wonderful. I'm glad I finally figured out the music so that we could do one song together. You guys are awesome. Thank you very much for joining me. Have a great afternoon. Everybody stay safe. The social isolation that our leaders are asking us to do is really important. I mean, this is all biology. This is all virus. We do not want this virus to spread. The consequence of it spreading is breakdown of hospital systems, right? And we don't want that to have happen. So as hard as it is, maintain physical distance, connect like this, and ultimately our society, our culture will beat this virus. It's going to take a long time, but I want to see you guys on Thursday. Stay healthy. You guys are the best. Thank you guys. I'm going to sign off. Take care of yourself, and I'll see you at the next video. And remember, the BIOMANIA app, please download it, and then it's $9.99 for an in-app purchase that unlocks all the content, and $19 to get access to this entire website. I think you'll agree that it's a fantastic deal. Guys, signing off. Take care. Stay safe. Mr. W signing